Biochemistry. Welcome to Biochemistry session, the first session for 2023 batch. So to begin with, why do we read biochemistry? So biochemistry is nothing but whatever food you are going to consume, they are going to get it converted into certain molecules, and they are going to be helping for us to build some molecule to destroy some molecule. So how we are going to classify our biochemistry entirely based on our feeding status. So either we are going to be in the fed state, or I am going to be in the fasting state, or I am going to be in the starvation state. So மூணு type of இதுல தான் நம்ம இருக்க போறோம். ஓகேவா? So I hope everyone are comfortable with Tamil. If you anyone have the problem with discussing in Tamil, you can just put it in the chat box. I'll try maximum to avoid Tamil, but sometimes it is better to explain you guys in your mother tongue. So that is the reason I'm just sometimes I'll be like unknowingly using Tamil words. So please adjust. So first thing is we are going to see how we are going to be how our metabolic reactions are going to take place when we are in the fed state. சாப்பிடும் போது சாப்பிட்டதுக்கு அப்புறம் ஏற்படக்கூடிய மாற்றங்கள், the changes that are going to be taken place is called as your fed state. Second thing is going to be your fasting state. Fasting எப்ப சொல்வோம், fed state எப்ப சொல்வோம். So இதெல்லாம் தான் basics before you jump into a metabolism. Fed state அப்படின்னா 2 hours. So after consuming your food, 2 hours it takes for our body to break down that food and separate it into different molecules like glucose, amino acids, and your fatty acid. In the my breakdown பண்றதுக்கு you require 2 hours. So on the 2 hours period, now we are going to call it as a fed state. Second thing is going to be your fasting state. So fasting state neppa solrona 12 to 18 hours after consuming a food. If you are not going to take food in between, you are going to be called as fasting state. So what is going to happen at this state? So whenever you're going to consume food, your body's requirement is taken from the food itself. It is not going to touch your reservoir. For example, if you're going to have a 10 rupees in your purse, you're not going to go and ask your mother for money, right? So you have your purse where you're going to have your money. Or in other words, ATM card la poi, if you want to take money, only after your purse is going to become empty, you're going to go to the ATM. So purse gets empty after 12 hours. In that 12 to 18 hours, what happens is you're going to go to a ATM, you're going to withdraw money using your ATM card, or you're going to go to your mom and you're going to ask money. So that part of period, 12 to 18 hours, what happens? Whatever storage product present in our body, for example, the immediate reservoir is going to be your glucose. So glucose is going to be stored in our body as glycogen. So that glycogen is going to get broken down to give you the food. Or next, what happens? Either you can go and take from your ATM, or else if you're going to be very brilliant, if you're going to be very criminal in nature, what you can do is you can print your money directly, right? So like how Simbu does in a movie, Poda Podi movie, he himself will be like printing money, right? Same way, you can go and print your glucose directly. So that is called as gluconeogenesis, newly producing your glucose. So that occurs in the fasting state. So now what happens? After 18 hours, after 24 hours, what happens is your body is not going to have glucose reservoir. Your bank account is also going to get emptied. Now you don't have money to withdraw from your ATM also. Now what you have to do is you have to go and lend money from others. So that's how your body is going to lend your ATP glucose from your fatty acid, from your protein. Now what our body does is he goes and tells to fatty acid which are getting stored in our body, proteins which are getting stored in our body, and goes and tells that, dude, I'm not having a money right now. I'm not having a glucose right now for utilization of energy. So now what you're going to do is you're going to help me by lending. So 1 to 3 days, like he goes and asks your near close friend. If close friend is also not giving, that is your fatty acid is also not giving, now he is going to go to some other third person and asking for the glucose molecule. So now what happens? In the prolonged starvation, after 3 to 5 days, there is going to be a protein breakdown. That is going to be your amino acid. That amino acid is going to join your cycles and it is going to produce ATP. Ultimately, why we are depending on glucose? for ATP molecules. So that is exactly entire biochemistry summarizes, guys. So if you're going to understand this, you will be like understanding entire biochemistry. I'll give you 4 important rules or 4 important formulas today. Okay, in the basic concepts, you're going to remember 4 important formulas. The first formula is you need to understand. You're going to read lots and lots of cycle in the next few weeks where we are going to discuss biochemistry. So you're going to discuss various number of pathways which are going to study in your biochemistry. If you want to understand that pathway, you are going to classify broadly into anabolic pathway and catabolic pathway. In anabolic pathway, everything is going to get building up. So if you're going to have excess of glucose, they need to store it as glycogen. If you're okay, so that glycogen storage is going to be called as glycogenesis. Then you have HMP shunt pathway where it is going to store the glucose as some other molecule, which we will be discussing. Then you're going to have your synthesis of everything. So anabolism literally means production. Catabolism literally means destruction. So fatty acid production, cholesterol production, triglyceride production, all these are going to come under anabolic pathway. And the last and final thing you need to remember is going to be your lipoprotein lipase. Lipoprotein lipase. Now, what is this lipase? Lipase means, as the word tells you, lipid ACE. So ACE means breaking down. So there is a substance called as lipase enzyme, which is going to break down your lipids to produce a energy. அப்ப அந்த லிப்பிட் ப்ரொடக்ஷன் லிப்பிட பிரேக் டவுன் பண்றது இருக்கு பாத்தீங்களா லைபேஸ் இது it occurs both in the anabolic pathway as well as catabolic pathway. When you're going to read your directly your fatty acid metabolism, you will be confused. Lipid metabolism direct up போகும்போது you will be literally confused why lipase is present in catabolic as well as anabolic. Guys, I will tell you now in the basics itself. Okay, so lipoprotein lipase is a anabolic pathway, whereas in catabolic pathway you're going to have another one called as hormone sensitive lipase. Hormone sensitive lipase. So one more thing I would like to tell you is copy down whatever I am discussing. Study in this order itself. Even while going to your university exam, read in this order. You will be like excelling well in your exam. Don't read like lipid metabolism, carbohydrate, integrate and learn. So my basic discussions will be entirely integration of each and every pathway into one single thing. So you will be observing it in the upcoming days. Okay. So lipoprotein lipase is going to be there. So and then you have hormone sensitive lipase. So remember, lipoprotein lipase is going to be anabolic pathway, whereas your hormone sensitive lipase is catabolic pathway. Now coming to the other catabolic pathways, you have everything lysis, glycolysis, glycogenolysis. You're going to just break down all the products. So you're going to have your beta oxidation of fatty acid. So you're going to have gluconeogenesis. Okay, remember gluconeogenesis is also a catabolic pathway. Then you're going to have your ketone body synthesis. So ketone body utilization. Now can you observe one thing? All this catabolic pathway are going to be present in which state? In your fasting state or your starvation state. In all these things, you're going to have your catabolic pathway. You're going to have catabolic pathway. Whereas fit state, you're going to have anabolic pathways. Anabolic pathways. So that is ultimate concept which you need to understand. Now coming towards the SMILE formula 1. Here you're going to differentiate between the two lipase, lipoprotein lipase and your hormone sensitive lipase. Lipoprotein lipase na enna. So for example, if you're going to see, you're going to eat a fat. This fat is going to get converted into triglycerides in your intestine. So your triglycerides are going to get absorbed from your intestine. Then what happens is, how to solve, definitely I'll be telling you, don't worry, and I will be discussing. So that's why I'm going concept wise. So first understand these 4 formulas that will help you to solve all your cases. Any case given in your exam, you will be able to differentiate just by using these 4 formulas. The first formula is you need to differentiate, is it an anabolic pathway or catabolic pathway? Anabolic pathway na within first 2 hours. That is going to be just anabolic pathway after eating food. Remaining everything is going to be your catabolic pathway. Now I'll differentiate your lipoprotein lipase and hormone sensitive lipase. See, so after eating food, you're going to have your triglyceride. This triglyceride is going to be attached with chylomicron. They are going to be attached with a molecule, a transporter of your triglyceride called as chylomicron. So this triglyceride and chylomicron is going to go in your blood circulation. This is going to be your blood vessel. But what happens in your endothelium of blood vessel? I hope you know what is endothelium. That is going to be the first layer of your blood vessels. In your endothelium of your blood vessel, you are going to have lipoprotein lipase. Your lipoprotein lipase is going to be present in between the two endothelial cells. This lipoprotein lipase is going to come and act on this triglyceride. Now, what happens whenever you're going to break down your triglyceride, you're going to get three fatty acid molecule plus a glycerol. So, three fatty acid plus glycerol is going to be the triglyceride composition. Now, what happens? This free fatty acid and triglyceride is now going to enter into your adipose tissue. Now, this is going to be your adipose tissue. Okay, so now here it is going to break down your triglyceride accepted. So you may consider it as a catabolic reaction. But what happens? The broken down three fatty acid and glycerol is going to enter your adipose tissue, where they are again converted into your triglycerol and they are going to get stored. That is the reason why we call this as a anabolic reaction. So you're going to store your triglyceride in your cell, in your adipose tissue, and that is the reason you're going to call it as an anabolic reaction. Now coming to the role of your catabolic reaction of your hormone-sensitive lipase. What happens in your hormone-sensitive lipase? So now you're going to have your adipose tissue. In your adipose tissue, you're going to have your triglyceride. This triglyceride will be converted to your three fatty acid and your glycerol once again by hormone sensitive lipase. Here also it is going to it is going to break down your triglyceride, but where is it acting? It is going to act on your adipose tissue. So whenever it is going to act on your adipose tissue, it is going to break down your stored products. Whenever you're going to break down your stored products, that is going to be your catabolic reaction. So here it is good. Now this is going to enter into your blood. It is going to enter into your liver. This is nothing but your beta oxidation of fatty acid. So it is going to convert into a beta oxidation of fatty acid to provide ATP. Okay, now coming to the second SMILE formula. Are you all following with me, guys? Any doubt? No, right? Yes. Now coming towards the second formula that is going to deal with the hormones. Now you're going to deal with something called as hormones. So remember, ultimately entire metabolism is going to depend on one important hormone, that is going to be your insulin. Okay, that is going to be your insulin. When is your insulin is going to get produced? I'll tell you the simple thing which everyone doesn't know. Okay, maximum number of people, your seniors might also not know this concept. Listen carefully. So you are going to eat a food. Okay, you're going to eat a food that is going to be a carbohydrate. Now, this carbohydrate gets broken down into glucose, monosaccharide. This glucose is going to get absorbed by your small intestine. Now, that is going to enter your blood vessel. Now, your blood vessel is going to consist of glucose. Okay, now this glucose is going to enter into your pancreas. So, now you're going to have your pancreas. This pancreas consists of something called as beta cells. So this beta cell is where your glucose is going to enter. Now remember, for the entry of glucose in your peripheral tissues, in your muscles, you require something called as transporter of your glucose. But your beta cells doesn't require transporter of glucose. So what happens? The glucose which you are going to consume is going to now enter into the beta cell. Now, whenever a glucose enter into a cell, this glucose is going to undergo glycolysis. Okay, it is going to undergo glycolysis. This glycolysis will be producing something called as ATP. This ATP is going to cause opening of potassium channel. Opening of potassium channel inside your, I hope so you all well versed with the general physiology which is given in your Guyton, in your physiology, right? So you have to know what is a potassium channel, what is going to be a voltage gated sodium channel, ligand gated sodium channel. I hope you all know. If not, you can go to my YouTube channel, Medicine, where we have general physiology completely covered as a video lecture. You can just go and see that. Okay, so now coming back to your this concept. So while opening of potassium channel, what happens is when the potassium channel gets opened, it is going to cause what? Anyone? So whenever the potassium channel is going to open, it is going to produce insulin. So the beta cells are going to produce insulin whenever there is going to be ATP. Now the question comes, if you are going to be asked which is going to cause the synthesis of insulin, many people will tell it's a glucose or a monosaccharide which causes production of insulin in your beta cells of pancreas. But if you are going to be a topper, if you are going to be a standard of USMLE, then you need to tell that it is not glucose that is going to open up your potassium channel and produce insulin in your beta cell. It is going to be the ATP which is going to produce insulin. Okay, so you need to be very well careful. So this actually I want to give a credit here. So this was discussed one and only by my faculty. Now he is in Meenakshi Medical College. If anyone from here is in Meenakshi Medical College, you might know him, Dr. Desigamani. So he is an excellent biochemist. He was the one who actually opened my eye in this concept. So every textbook gives you that it is going to be the glucose that is going to be producing insulin, but it is not the glucose that produces insulin, but it is going to be the ATP that is producing insulin. Okay, if anyone sees him, if anyone from here is from Meenakshi Medical College, do go and ask him this question. He will be telling you this concept very well. Okay, so now coming back to your insulin. So insulin is released at what state now? It is going to be released in the fit state. Now, can you tell me, is it an anabolic hormone or catabolic hormone? It is released in the fit state. So can anyone put it in the chat box? Is it an anabolic hormone or catabolic hormone? Your insulin. Can you put in the chat box? Come on, guys. Very good, Darsh. Any other? So kindly put in the chat box, guys, whenever I'm asking. That gives you a practice to apply whatever you're reading, okay? So this is a place where you can start with applying whatever you're listening and whatever you're reading. Excellent. It is going to be your anabolic hormone. It is an anabolic hormone. Very good, Darshan, Anis. Okay, now coming to the next one. You're going to have this next hormone that is going to be your glucagon. Glucagon. Okay, this glucagon is going to be present in the fasting state and starvation state. Now, can you tell me what type of hormone it is? Anabolic or catabolic? Now, can you tell me, is it anabolic or catabolic? Your glucogen or okay, your glucagon. Very good. It is going to be catabolic in nature. Excellent. So remember, insulin activates all the anabolic pathway. Okay. So insulin is going to activate all your anabolic pathway. Okay. All anabolic pathway is going to be activated by your insulin. Anabolic pathway is going to activate by your insulin. Now you may ask me, your glycolysis is happening. Without glycolysis is going to be a catabolic pathway, isn't it? Now you will ask me, it is activating only anabolic pathway, but glycolysis is a catabolic pathway. Excellent question. That is C. So all anabolic pathways activated by your insulin, as well as two catabolic pathways activated by your insulin. What are the two catabolic pathway activated by your insulin? Number one, it is going to be your glycolysis. And number two, it is going to be your link reaction. I'll tell what is this link reaction, guys. It is called as link reaction. We will be discussing about that. Don't worry. So two things. One is going to be your glycolysis. Another is going to be your link reaction. Both are going to be activated by your insulin. Now you will ask me the next question. Your glucagon is going to activate the catabolic hormones. So what are the catabolic things? Remember, all catabolic pathway is going to be activated by your glucagon, except these two. So except your glycolysis and your link reaction, which is activated actually by your insulin. Clear, right? Now coming to this important thing. So remember, you have various hormones in your body. You will be reading all these hormones while you're going to discuss your endocrine physiology in your physiology. So your glucagon, growth hormone, glucocorticoid, epinephrine, norepinephrine, your thyroid, everything is going to increase your blood glucose except your insulin, which will be decreasing your blood glucose level. Okay, what happens is all these glucagon, growth hormone, glucocorticoid, எப்படி increase பண்ணதுன்னா, they are going to activate your adenyl cyclase. Remember, whenever your ATP is going to be there, It needs someone to activate, isn't it? So whenever there is going to be a activation of ATP, who requires for activation of ATP? You need glucagon, growth hormone, glucocorticoid, epinephrine, norepinephrine, and thyroid. So now what happens? All these things are going to decrease the ATP level in your body by converting your ATP into cyclic AMP. So that is why breaking down of ATP is occurring. Therefore, you're going to call it as a catabolic hormones. So how it is going to be occurring? Because of your adenyl cyclase. Now, whenever your adenyl cyclase is going to be present, what your adenyl cyclase is doing is, your adenyl cyclase causes phosphorylation. Your adenyl cyclase is going to cause enzyme phosphorylation. So you have different enzymes and different metabolic pathways, isn't it, guys? For activation of all these metabolic pathways, you require phosphorylation of enzyme. Okay, so this phosphorylation of enzyme is done by all your catabolic hormones. Okay, so now how your insulin is decreasing? I'll tell you guys, your insulin is going to activate your phosphodiesterase. Now what happens? Your cyclic AMP, as soon as it is getting converted, your cyclic AMP is going to completely getting destroyed into 5-AMP. So when they are going to destroy it into a 5-AMP, will you have protein kinase activation? No. So once you don't have the protein kinase activation, there will be no phosphorylation. So now can you tell me which causes enzyme phosphorylation? Which one does it cause? Which ones causes phosphorylation? Whether it is going to be your glucagon or whether your insulin which causes phosphorylation, in the chat box, please. Whether it is going to be your insulin or your glucagon which causes phosphorylation. Phosphorylation of enzyme, very good. It is going to be your glucagon. So remember the formula 3 which we are going to discuss is, number 1, the pathway which is activated. I want you all to write it down, guys. Pathway which is activated by insulin will be active at dephosphorylated state. For example, can you tell me what are the pathways activated by your insulin in the chat box? What are the pathways that are getting activated by your insulin, guys? What are the pathways that gets activated by your insulin in the chat box, please? Each one, can you give me one in the chat box? What are the pathways that are going to get activated by your insulin? Very good. Glycogen synthesis. Is it a glycogen synthesis? It's called as glyconeogenesis. Okay. Or glycogenesis. Put it as glycogenesis. Okay. Va? So glycogen synthesis taniya sollakudadu. Always mentioned as glycogenesis. Excellent. Glycolysis link reaction. What are the other things? You have, what are the other synthesis you do in your body? What are the other components that are going to be synthesized in your body? Your fatty acid synthesis, you have triglyceride synthesis, you have cholesterol synthesis, you have HMP pathway. All these are going to be in the dephosphorylated state. So they are going to be activated at the dephosphorylated state, whereas the pathways which is going to get activated by your glucagon will be active at which one? At phosphorylated state. Okay. So this is going to be the third formula in your basics, which you are going to remember. Now coming to the formula 4. What is that formula 4? Remember, all your anabolic pathways, all your anabolic pathways are going to be occurring in your cytoplasm. All your catabolic pathway is going to occur in the mitochondria. So there is one exception to this mitochondria. There is only one exception to this. What is that exception is? You know, your glycogenolysis is there, right? So smile formula 3. Yeah. So the pathway which is going to get activated by your insulin, all your pathways, all your anabolic pathways is there, right? So they are going to be activated at dephosphorylated state. So you have tyrosine kinase enzyme, guys. This tyrosine kinase enzyme will be phosphorylating your enzymes. At phosphorylated state, you have all your catabolic pathway. And in your dephosphorylated state, without the action of your protein kinase A, there are some activation of pathway. They are going to be anabolic pathway. That is why your insulin is going to activate your anabolic pathway, whereas your glucagon and all these hormones are going to activate your catabolic pathway. Okay, so that is all about your SMILE Formula 3, and I hope Mahalakshmi is clear with the concept. Now we are going to move towards SMILE pathway 4. So all the anabolic pathway are going to be present in the cytoplasm, and all your catabolic pathway is present in your mitochondria. Now the thing is, there are some exception. So what is that exception? Okay, well, can you tell me what is your glycogenolysis? Is glycogenolysis a catabolic pathway or anabolic pathway? Glycogenolysis. Glycogenolysis. Is glycogenolysis a anabolic pathway or catabolic pathway? Can you put in the chat box? Glycogenolysis. Is it anabolic pathway or is it catabolic pathway? Anyone? Yes, it's a catabolic pathway. As per the formula, it must be in the mitochondria. But remember, your glycogenolysis is okay, it is going to be starting, it is going to be occurring in your cytoplasm. So if instead of remembering each and every pathway where it is occurring, you can remember like this: all your anabolic pathway which you are reading in your biochemistry, they are going to be occurring in your cytoplasm, and all your catabolic pathway occurs in your mitochondria. There is some exception like your glycogenolysis, which is going to be occurring in your cytoplasm. Now, you have to remember another exception. There are some pathways which occurs both in mitochondria and cytoplasm. That means that some starts in your mitochondria. Okay, some pathway is going to start in your mitochondria, but they are going to get continued in your cytoplasm. What are those pathways? Number one is going to be a urea cycle. Number one is going to be a urea cycle. Number two is going to be your heme synthesis. Number two is going to be your heme synthesis. And number three is going to be your glucogenesis. Glucogenesis. So all these three, they are going to be starting in your mitochondria. They are going to start at mitochondria, but they are going to come to your cytoplasm for completion of pathway. So that you're going to remember. So with this SMILE Formula 4, you can just remember the place where. So don't worry, I'll just summarize at the end, Janet, or else also you can just see that in the YouTube. I'll upload it in the YouTube. Don't worry. But it will take 1 or 2 days maximum. I'll upload in the YouTube. That time you can see, because I'll try to update, get a quick summary at the end of the session, not now, because it will be affecting the others, those who are seeing the session, right? Okay, don't worry, Janet. I'll just try to summarize it. Okay, so now coming towards the other thing. So you have some pathways that is occurring in your cytoplasm, some pathways occurring in the mitochondria. So I have already told you. So your HMP shunt pathway is going to be your anabolic pathway. Glycogenesis is going to be anabolic pathway. Okay, fatty acid synthesis is anabolic pathway. Cholesterol is anabolic pathway. So exception is going to be your glycolysis and glycogenesis, glycogenolysis, which are actually catabolic, but they are going to be present in the cytoplasm. Now coming towards your mitochondria, you have all your catabolic pathways, like all the beta oxidation of fatty acid, link reaction. You have your vital pathway like your TCA cycle. Everything is going to be from your mitochondria. It is not going to be from your cytoplasm. And I hope so. Now you will be understanding the location of all the pathways, whichever we are going to discuss later on in your biochemistry. Now coming to the last one. Okay, now we are going to come to the last one. So before moving to that, let's go into some MCQs, guys. Let's move into some MCQs. Okay, so before moving into that, you know the two lipases too, right? So now let's go into the MCQs. Let me see who is going to answer the first question. Which of the following is active in dephosphorylated state? Can you guys answer in the chat box? For that, you need to understand which is going to be anabolic pathway and which are going to be in the catabolic pathway. So I will tell you guys, glycogen synthase. The word itself, now you don't know any biochemistry, okay? So now we are going to start. We don't know any biochemistry. So I will tell you how to answer. Synthesis. So since it's going to have synthase, it is going to be synthesis. So synthesis means it is going to be anabolic reaction. So remember your pyruvate carboxylase. Anyway, we are going to discuss it later, but for now, just write it in your revision note. So this revision note, whichever you're copying it from my sessions, it will be useful for you at the end of the year before your final year exam. Okay, your pyruvate carboxylase, it is going to be used in the gluconeogenesis. Can you tell me your gluconeogenesis, is it a catabolic pathway or anabolic pathway? Gluconeogenesis, is it a catabolic pathway or anabolic pathway? Can you put in the chat box? Gluconeogenesis. 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 It's a catabolic pathway or anabolic pathway? Gluconeogenesis. Everyone, some are telling anabolic pathway. Let me help you out. So for this, that's why I've just given this option. See, gluconeogenesis. When will you generate glucose in your body? Is it in the fed state or is it in the starvation state or fasting state? When will you generate glucose in your body? When will you generate glucose in your body? In the fasting state. I told you guys that fasting state, starvation state, prolonged starvation state. Even எல்லாருமே they are going to come to the catabolic sum. They are not going to be anabolic stage. They are going to be a catabolic reaction that is going to be taking place in our body. So remember, your gluconeogenesis is also occurring in the fasting state. Therefore, they are not going to be anabolic. Don't get confused with the terminology genesis. Remember, gluconeogenesis is the catabolic state. Why? Because it is going to be occurring in your fasting state. So therefore, this is going to be a catabolic reaction. Now, glycogen phosphorylase. Phosphorylase. So it is going to be having the terminology phosphorylation. Now tell me, when will you have phosphorylation? Only in your anabolic or catabolic reaction? Phosphorylation occurs in anabolic reaction or catabolic reaction? Guys, come on. Put in the chat box. Phosphorylation occurs in the anabolic reaction or catabolic reaction? Phosphorylated in the catabolic reaction. So it is going to be a breaking down of glycogen. So it is going to be a breaking down of glycogen. And now coming to the next one, acetyl-CoA carboxylase. Remember, this is going to be present in the fatty acid synthesis. So now, for now, write it down. You don't know this. So acetyl-CoA carboxylase is going to be an enzyme used in your fatty acid synthesis. Now, is it an anabolic or catabolic reaction? This you all know. Fatty acid synthesis. Is it anabolic or catabolic? When will you synthesis fatty acid? It's an anabolic reaction. There's going to be anabolic reaction. It's not a catabolic reaction. Now coming to your pyruvate dehydrogenase. Again, this is present in your link reaction. Your link reaction is going to be a catabolic state. But remember, even though it is going to be a catabolic state, your insulin will be acting on two exclusion in your catabolic state. That is going to be your glycolysis and your link reaction. Therefore, even though your pyruvate dehydrogenase, which is present in your link reaction, is a catabolic hormone, okay, catabolic enzyme, insulin is going to activate your link reaction. Therefore, it is going to be a dephosphorylated state. So remember, your insulin activates only your enzymes that can be in the dephosphorylated state. The enzymes that are active in the dephosphorylated state. So now can you tell me, out of these 5 options, which are going to be activated by your insulin? Out of these 5 options, which are going to be activated by your insulin? Out of these 5 options, which are going to get activated by your insulin? Option A, will it be activated? Yes, of course it will be activated. Option C, again option D, again it is going to be a anabolic. All your anabolic reactions are activated by your insulin. Apart from that, 2 catabolic reactions are activated by your insulin. So that is what we were discussing in the SMILE formula. Okay, that is going to be, we have discussed in your SMILE formula 3. Okay, so SMILE formula 3. So it is going to be your link reaction or pyruvate dehydrogenase. So here the option is going to be A, D, and E. Now coming to the second one. Come on, fast. Which of the following does not occur in the mitochondria? Which of the following does not occur in the mitochondria? So remember, in your mitochondria, it is going to be your catabolic reaction except your glycolysis and your glycogenolysis. Now can you tell me, out of this, which does not occur in the mitochondria? So this is going to be an anabolic reaction. Therefore, fatty acid synthesis does not occur in your mitochondria. Excellent. Ziomi, please rename yourself so that I can understand your name instead of calling you as Ziomi. And Kiran Kumar, thank you. You also answered it correctly. Very good. Now coming to the third one. Insulin promotes lipogenesis by all except. This is your third question. Lipogenesis. So insulin is a anabolic hormone. First thing you can understand from your question is insulin is an anabolic hormone. Now they are asking except. Okay, they are going to ask except. So will it decrease the cyclic AMP? Option A is correct or wrong? Yes. So they are going to decrease your cyclic AMP by activating your phosphodiesterase. By activating your phosphodiesterase. Therefore, option A is going to be true. Now coming to the option B, will it increase the glucose uptake? Of course, yes. They are going to increase the glucose uptake in your peripheral tissues. So answer is 2. Now, third thing, it is going to inhibit pyruvate dehydrogenase. Just I discussed that your pyruvate dehydrogenase is going to be a link reaction. If it is going to be a link reaction, will it inhibit? No, it is going to activate. It is going to activate your pyruvate dehydrogenase. So this is going to be a false, and it increases your acetyl CoA. Of course, yes. So this is going to be also true. Okay, wow. So how this is going to be occurring? This is your link reaction. Pyruvate, in the presence of pyruvate dehydrogenase, it is going to get converted into acetyl CoA. So it is going to get converted into acetyl CoA. So this is your link reaction. So it is going to activate your insulin. Insulin is going to activate your link reaction. So whenever we are going to discuss your carbohydrate metabolism, you should remember this step. Okay, so we are going to go layer by layer. Understanding the basics first is very important, and that is what I am exactly doing here. Now coming to the fourth one. Mitochondria are involved in all except. Can I put in the chat box, guys? Mitochondria are involved in all except. Mitochondria is involved in all except. Is it involved in your ATP synthesis? Is mitochondria involved in your ATP synthesis? Of course, yes. So why we are just breaking down your glycogenolysis or your gluconeogenesis? Everything is going to be for ATP production only. So ATP production is of course going to be involved. So ATP production is of course going to occur. Next, coming to the B, option B, apoptosis. So remember your mitochondria, this you will be learning next year, but now you remember this. Apoptosis is going to be a programmed cell death. Programmed cell death is going to be occurring in your mitochondria. That is also true. Tricarboxylic acid, TCA cycle is also occurring in your mitochondria. True. But your cholesterol synthesis is going to be a anabolic reaction. Anabolic reaction does not occur in your mitochondria. So this is going to be a false, and option D is the correct answer. And very good, Praniti and Kiran Kumar. Excellent. So coming to the option, question number 5. Hormone sensitive lipase is not activated by. Who is going to answer this? Hormone sensitive lipase, whether it is anabolic reaction or catabolic reaction. It is going to be a catabolic reaction. So insulin does not activate your hormone sensitive lipase. Coming to the sixth question, which of the following is not seen in the low insulin glucagon ratio? Low insulin glucagon ratio. So insulin divided by glucagon is going to be low. Upper, which is going to be high. Okay, when this is going to be low, your insulin will be low or your glucagon is going to be high. So when the glucagon is high, that means that it is going to be a catabolic reaction. Out of this 4, which is going to be not a catabolic reaction. Now they are asking which of the following is not a catabolic reaction. Can you tell me? Is gluconeogenesis anabolic or catabolic? It's going to be a catabolic. Glycogen breakdown, it's a catabolic. Ketogenesis is a catabolic, but glycogen storage or glycogenesis, it is not going to be a catabolic reaction. So glycogenesis, okay, is going to be a anabolic reaction. Therefore, it's going to be option A, anabolic reaction. Option D, glycogen storage. Now coming to the next one, which of the following is active in dephosphorylated state? So again, there's going to be another one question. Let me see who is going to answer this. Synthase. So glycogen synthase, it is going to be anabolic reaction. Therefore, this is going to be the right answer. Remember, pyruvate carboxylase is going to be present in gluconeogenesis. So that is a catabolic reaction. Glycogen phosphorylase, okay, it's going to be glycogenolysis. So that is also your catabolic reaction, and we will discuss about this PEPCK. Don't worry for now. Okay, next one. All occur in mitochondria except. Next, let me see who is going to answer this question. All occurs in the mitochondria except. Who is going to answer this? All occurs in the mitochondria except. will glycolysis occur in the mitochondria? No. Glycolysis occurs in the cytoplasm, guys. That's what I told you. Only catabolic reaction that is going to be occurring in your cytoplasm is going to be your glycolysis. So that is the option A is correct. TCA cycle occurs in the mitochondria. Ketogenesis. Don't get confused with this word genesis. Ketone body synthesis agudinale. So that is going to be a catabolic reaction. So ketone bodies are going to be synthesized only when we don't have glucose for consumption. So a ketone body ever produce in the fasting state or prolonged fasting state. So that is a catabolic state. So this is also going to be a catabolic reaction. And ETC cycle, again, it is going to be occurring. It's going to be a vital reaction that is going to be occurring in your mitochondria and not cytoplasm. Next, coming to the 9th question, biosynthesis of enzyme pyruvate carboxylase is repressed by. So carboxylase, pyruvate carboxylase, already we saw it is going to be present in which reaction? Pyruvate carboxylase. So pyruvate carboxylase we saw in case of gluconeogenesis. So pyruvate carboxylase occurs in the gluconeogenesis. So this is going to be gluconeogenesis. So can you tell me which is going to repress your gluconeogenesis? Which is going to repress your gluconeogenesis? Can you someone put in the chat box? Can someone put in the chat box which is going to repress gluconeogenesis? So it is going to be your insulin. Very good, very good. Now you are all coming back to the form. Okay, you can, if you're going to read like this, you will definitely develop an interest, guys. Next, which of the following is active in phosphorylated state? Phosphorylated state, na catabolic reaction. Now, out of this 4, which is going to be catabolic reaction? Glycogen synthase, glycogenesis. So it is going to be an anabolic reaction. Glycogen phosphorylase. So glycogenolysis. It is going to be a catabolic reaction. Acetyl-CoA carboxylase. It is going to be an enzyme involved in your fatty acid synthesis. So it's anabolic reaction. G6PD enzyme is going to be an enzyme in your HMP shunt pathway. Therefore, it is also going to be an anabolic reaction. Therefore, remember option B is the right answer. Now, someone asked me in the chat box how to approach a question, a clinical case-based questions in the exam. So that also I'll be discussing in between every topic. Don't worry. Now coming to this question, a professional cyclist has been preparing for a long race competition. To guarantee that muscle glycogen is stored after the workout, his coach strongly advises him to consume carbs. That is going to be a carbohydrate. Which of the following is most likely cause an increase in the activity of rate limiting enzyme of muscle glycogenesis under such activating state? So simple, muscle glycogenesis occurs அப்படின்னா glycogenesis. When do the muscle glycogenesis occurs? Muscle glycogenesis is an anabolic reaction. So if it is an anabolic reaction, okay, if it is an anabolic reaction, what is going to be the enzyme involved? It is going to be insulin that is involved. Now tell me whether insulin out of this 4, what is the best answer for insulin? What is the best answer for insulin? Can you put in the from out of this 4? It is going to be a dephosphorylation. So remember, so which of the following most likely cause an increase in the activity? Dephosphorylation caused by your insulin will be causing rate limiting enzymes of muscle glycogenesis. Now the question comes, a simple question. Sir, my patient is going to be a diabetic in nature. So diabetes Nariya glucose is present in the blood vessel. That is the problem for them. So they are not glucose deficient, but there is excessive of glucose. Now tell me why there is a complication. When will you have a complication? When these glucose are going to form a AGE product. That is going to be your advanced glycation end product. So whenever your glucose, so either I'm talking about a diabetic mellitus patient. In diabetic mellitus patient, so there is a excess of glucose present in your blood. So these are not going to be taken up by your muscles because why? Because they don't have this. Because they don't have this. Can someone tell me? You don't have the insulin which is required for your transporter, blood transporter, okay, which is going to be GLUT4 transporter, which is going to be present in your muscles. So they require insulin. Now what happens is when you're then why you are asking the patient to run, patient to do exercise? Anyone? Remember, why you are asking the patients of diabetes to do exercises? These glucose can be utilized. These insulins, these glucose can be utilized by your muscles. Okay, these can be utilized by your muscles without the requirement, without your requirement of insulin. by activation of GLUT2 receptors when it is going to be in the active state. When it is going to be in the active state. Active muscle contraction நடக்கும்போது, active muscle contraction நடக்கும்போது, you don't require GLUT4 transporter which is dependent on insulin. Instead, it can go in a insulin independent absorption. Insulin independent pathway can get activated. So that is why you're asking the diabetic patient to run. Now the diabetic patient nowadays they are seeing Google and coming to you. So they will be asking you, sir, anyway muscles are having the GLUT4 transporter. This GLUT4 is a insulin dependent. For me the problem is insulin. So instead of running or doing a exercise, I can just put insulin, right? So that is ultimately required. No, you need to explain to the patient that it is not just giving insulin going to help you. You wanted to shift your muscles from insulin dependent blood transporter to insulin independent blood transporter. Therefore, if the patient is going to do a exercise, so now what happens? Many patient comes to you, sir, na morning ஒரு 5 kilometer walking போறேன், 6 kilometer walking போறேன். எதுக்கு walking போறாங்க? Useless, because while you're going to go for a walking, it is not going to cause contraction of your muscles effectively. Instead of going for 5 kilometer walking, they can go for 2 kilometer jogging, or they can go 1 kilometer running. That is equivalent enough to absorb all the glucose that is going to be present in your blood, which is going to be utilized now by your muscles without the insulin dependency. So this is an important concept I want you all to remember. Now coming back to this question, so here the rate limiting enzyme is going to be your insulin that is going to playing a major role. Your insulin is going to cause dephosphorylation of enzyme, and dephosphorylation of enzyme will be helping you for absorption in your muscles, and they are going to be helping you. So in a normal patient, insulin will be there, no worries. So they are going to help you in the dephosphorylation. And dephosphorylation ஆச்சுனா என்ன ஆகும்? நம்ம பாடியில glucose effectively reduces. அதனால தான் after the workout, the coach is advising you to consume some carbohydrate, because now what happens? The blood glucose level will be decreased. Therefore, you need to go for a muscle carbohydrate. You have to eat carbohydrate so that the muscle glycogens is not going to be utilized. Glycogen will be not disturbed. So muscle glycogen will be stored even after the workout if you're going to consume carbohydrate. Clear, right? This is an important concept I want you all to remember, because you have lots of diabetic patients in your family also. Most of them will have. So they will ask these stupid questions to you guys. So you need to explain them and make them understand. Okay. That is the ultimate thing which we are going to do. Now, coming to the next one. Now, can you guys tell me what are the sources of blood glucose? What are the sources of blood glucose? Can you put in the chat box? What are the sources of blood glucose? So some people will have increased glucose, right, in their body. Can you tell me what is going to be the sources of blood glucose in your body? Anyone? Can you tell me what is the source of blood glucose in your body? What are the sources of blood glucose in your body? Come on, come on. Can you tell me what are the sources of blood glucose in your body? What are the sources? Number one, it is going to be your food. Number two, it is going to be your glycogen that is stored in your liver. So this is what I told you in the fed state, fasting state. So glycogen that is going to be stored in your liver. Third one is going to be your gluconeogenesis. இது மூணு மெத்தட்ல தான் நமக்கு குளுக்கோஸ் வந்து இன்க்ரீஸ் ஆகும் நம்மளோட பிளட்ல. So remember, fat, they can never get converted into glucose. Fat can never get converted into glucose. Fat can only give ATPs. For breaking down of fat will give you ATP, but breaking down of fat will never give you glucose. But reverse is possible. Glucose can get converted into fat and get stored, but fat cannot get broken down into glucose. This is a very important thing which you are going to remember. Okay, next thing is going to be second question for you all. Question number one, you all answered. Now question number two, which is going to be preferred fuel? So can you tell me which is going to be the preferred fuel for your body? Is it a carbohydrate or fat or protein? Which is a preferred fuel for your body or main fuel for your body? Can you put in the chat box? Which is going to be the main fuel for your body? Yes, fat. Okay, Mahalakshmi is telling fat. Ziomi is telling carbohydrate. Others? Remember, your carbohydrate is always preferred all over your body. If you're going to take it normally, it's going to be a carbohydrate followed by your fat, followed by your carbohydrate, followed by your fat, followed by your proteins, otherwise your amino acid. Okay, wow. So remember, there are two things. Okay, there are two exceptions. There are two exceptions. Okay, where your fat can never be converted. Remember, your fat can never be converted into carbohydrate. You have two exceptions basically. I'll tell you. So remember, I told you the statement, right? Fat can never be converted to glucose. I told you, right? There are again certain exceptions here. So write down the exception. There are two exceptions. One is going to be your glycerol. So whenever you're going to break down your fat, whenever you're going to break down your triglyceride, you're going to have what? You're going to have fatty acid and you're going to have glycerol. Three fatty acid and glycerol. This glycerol can actually get converted into acetophosphate. That is going to be by glycerol 3-phosphate dehydrogenase. அப்படிங்கிற enzyme presenceல you are going to have this dihydroacetophosphate and this is going to get converted into your glucose. This can be converted into your glucose. So that is the one thing you have to remember. And second thing is going to be your propionic acid. Second thing is going to be your propionic acid. So in your fatty acid, you're going to have something called as propionic acid. This propionic acid, okay, what happens is they can get converted into your propyl CoA, propanyl CoA, propionyl CoA. Propionyl CoA, that is going to be a 3 carbon chain. So propionyl CoA, this propionyl CoA can get converted into propionyl CoA carboxylase enzyme presence. Carboxylase, word என்ன சொல்லுது? Carboxylase. So it is going to give you a carbon. It is going to break down your propenyl CoA. Okay, it is going to break down your propenyl CoA. So propenyl you're going to have 3 carbon plus CoA நம்ம வந்து வெறும் கார்பனை மட்டும் விட்டு வைக்கிறோம். So that you have a 4 carbon compound. What is that 4 carbon compound you're going to get? D-methyl malonyl CoA. Malonyl CoA. D-methyl malonyl CoA. This D-methyl malonyl CoA can actually get in the presence of racemase enzyme. In the presence of racemase enzymes, what happens? This D-methyl malonyl CoA gets converted into a L-methyl malonyl CoA. So D-methyl, L-methyl. இங்க வந்து 4 carbon compound அப்படியே தான் இருக்கு. Just அதோட structure மட்டும் நம்ம மாத்துறோம். Whenever you're going to change just the structure, you're going to call that as a racemase enzyme. Racemase. They are going to just change the structure. It is not going to change the number of molecules in the compound. Okay, so you're going to change it into L-methyl malonyl CoA. Malonyl CoA. This L-methyl malonyl CoA. So in the conversion, propenyl CoA to this 3C plus C compound, that is D-methyl malonyl CoA, they require carbon dioxide. So you're going to add a carbon dioxide. So you require a carbon dioxide. And second thing, you need a vitamin B7 or biotin. Okay, similarly, your L-methyl malonyl CoA in the presence of vitamin B12 and in the presence of mutase enzyme, what mutase? Methyl malonyl CoA mutase enzyme. It is going to get converted into your succinyl CoA. This succinyl CoA is going to enter your TCA cycle. They can get converted into your oxaloacetic acid. Oxaloacetic acid can be converted into your glucose. So this is how you are going to have this entire concept of propenyl, how your fatty acid have this chance of converting into your glucose. Now you're going to have this, L-methyl malonyl CoA. Now you all will be reading, right? Whenever you're going to have a vitamin B12 deficiency, they will be telling that the patient will have a peripheral neuropathy. Vitamin B12 deficiency, you will have peripheral neuropathy in solvanga. Why do you have peripheral neuropathy? Because this L-methyl malonyl CoA, they are going to be neurotoxic in nature. So they are going to be neurotoxic in nature. This L-methyl malonyl CoA can actually go and cause peripheral neuropathy. So when in the absence of vitamin B12. So now this also only the 16 those who are attending my session will today know. If you're going to go and ask even your second year seniors, if you're going to go and ask, they don't know this concept. Why you're going to have your vitamin B12 neurotoxicity? யாருக்குமே தெரியாது. Okay, எல்லாருமே வேற reason சொல்லுவாங்க. Just they get confused. But the ultimate reason is L-methyl malonyl CoA has to get converted into succinyl CoA in the presence of vitamin B12. If vitamin B12 is not there, L-methyl malonyl CoA is going to become neurotoxic and they are going to cause peripheral neuropathy. Similarly, when excess amount of L-methyl CoA is going to be present, they cause methyl malonyl urea. methylmalonyl urea. They are going to cause methylmalonyl urea. Clear, right? So this is all about your very important point. Next, we are going to discuss about next important topic, that is going to be in the fasting state, what happens. Okay, what happens in the fasting state? So let me tell you what happens in the fasting state. So whenever you're going to fast, so you're not going to take food today, you're going to have some people will follow this Saturday fasting, some people will follow this Ekadasi fasting, some people will follow many of my friends will be following a Ramzan fasting. So during this fasting, what happens? Let me tell you guys. Whenever you're going to have a fasting, so what happens is, first in your adipose tissue, what happens in your adipose tissue? You're going to have activation of hormone sensitive lipase. This hormone sensitive lipase is going to go and act on the fatty acid. So now you have excessive amount of fatty acid. This excessive amount of fatty acid need to go to your blood, right? So this is your adipose tissue. Now your fatty acid is going to enter into your blood supply. So inside your blood, what happens is, this fatty acid which is going to come down, they are going to get beta oxidation. They are going to be a beta oxidation of fatty acid. This beta oxidation, what happens? They are going to release acetyl-CoA. So remember, in your beta oxidation, your fatty acid is going to get converted into acetyl-CoA. This acetyl-CoA, they are going to get converted into three important things. What are the three important things? Either there is going to be a TCA cycle, either they can go to form a ketone body, or they can form a gluconeogenesis. Gluconeogenesis. Okay, they are going to form gluconeogenesis. So whenever you're going to have a TCA cycle, what happens? The energy is going to be produced in the liver. You're going to have a production of ATP, of course, yes. Then you have ketone body, where you're going to have the vital organs are going to take this ketone body. I'll tell you what are the vital organs which is going to utilize ketone body. Then you have gluconeogenesis. So this acetyl-CoA is going to activate your gluconeogenesis, and your gluconeogenesis, it is going to now enter into your liver. In your liver, there is going to be your enzyme called as pyruvate carboxylase enzyme. This pyruvate carboxylase enzyme activation will cause gluconeogenesis. Remember, acetyl-CoA will never be glucogenic. What is glucogenic? That is going to be production of glucose. Remember, your so your acetyl-CoA will never increase the blood glucose level. Blood glucose level increase பண்ணாது பாடியில, அதாவது டயபெட்டிக்கா ஆக்காது. That means, so it is not going to cause a diabetic level of your glucose level. So it is going to just satisfy your organs for glucose during the fasting state. But acetyl-CoA will never cause increase in the blood glucose level beyond the state. Because this acetyl-CoA, they are going to get either utilized, or they are going to help in production of glucose directly. Okay, wow. So that glucose production which is going to occur is going to be just for satisfaction of your fasting state. It is not going to increase your glucose level excessively in your body. Now coming to the diabetic state. So what happens in your diabetes? So what exactly happens in your diabetes? Let's see. In diabetes, what happens? There is going to be a decrease, or there is going to be absence of your insulin. Because there's a decrease or absence of insulin, what happens? The glucose which is present in the blood is not going to get utilized by your peripheral tissues. Peripheral tissues will not utilize. Now, if the peripheral tissues are not going to utilize, what your peripheral tissues think? They are going to think that the body is present in the fasting state. Even though you're going to consume carbs you're going to consume. So your peripheral tissue will think that it is going to be in the fasting state. Since it is going to be in the fasting state, what happens? It is going to cause increase in the catabolic hormones. It is going to cause decrease in the anabolic hormone. If it is going to cause decrease in the anabolic hormone, there is a decrease in the lipoprotein lipase enzyme. There is going to be increase in the hormone sensitive lipase enzyme, increase in the lipolysis, increase in the beta oxidation of fatty acid. It is going to cause ketone body synthesis. So only anabolic thing in diabetes which can occur is going to be what happens when in your diabetes, especially in a chronic diabetes. In a chronic diabetes, what happens is there is going to be a anabolic hormone which is functioning. I told you in your diabetes, only your catabolic functions are going to increase, but only thing that is going to be occurring in the anabolic thing is going to be, so the excess of acetyl-CoA, they will be converted into a fat. Okay, they are going to get converted into fat by fatty acid synthesis. You all know that your fatty acid synthesis is going to be your catabolic reaction. But remember, in your chronic diabetes, it's anabolic reaction. Sorry, it's anabolic reaction. So remember, only anabolic reaction that is going to take place in diabetes is going to be your fatty acid synthesis. It is not going to be any other anabolic function pathways which is going to occur except your fatty acid synthesis. This is your all about your thing. Now, let me see who is going to answer this question. Let me see who is going to answer this question. A person frequently eating roadside food presents with vomiting. So, comes to the physician due to food poisoning. He was unable to eat or drink properly for the past 3 days. Blood examination still showed normal glucose level. Which of the following is the most important for maintenance of blood glucose? Now, this is going to be a starvation condition. There is going to be a starvation condition. In starvation condition, which is going to help you, can you put in the chat box, which is going to help you in maintaining the blood glucose level? In your liver, there is a process called as, can you tell me with this? Which one occurs in the liver, which is going to help you in maintenance of glucose? Gluconeogenesis. It is going to be gluconeogenesis. So what is the option here? So gluconeogenesis is right, but this gluconeogenesis occurs in your liver. So the option B is the right answer. Is it clear, everyone? Is this question clear to everyone? Right. Now moving on to discuss about this important. This is going to be your formula of 5. So I have discussed 4 formulas. This is going to be the formula of 5. Smile formula of 5. So which is going to discuss about the fuel used in different situation. Fuel used in different situation in your body. Situation in body. Okay, so in brain. So remember, while you are going to eat food properly, so what you're going to feed? Carbohydrate you're going to feed to your body. So whenever you're going to take carbohydrates, what happens? Your brain is going to utilize the carbohydrate. Your liver is going to utilize the carbohydrate. There is no problem. Your muscles are going to utilize the carbohydrate. Your adipose tissue will be utilizing carbohydrate. Your RBC will be utilizing carbohydrate. But the only problem is your heart is going to feed on fatty acid. Remember, your heart will always feed on fatty acid even if you're going to consume carbohydrate. So for the heart, the nutrition is going to be given by fatty acid. ATP is synthesized by fatty acid, not your glucose. So apart from that, everything is going to be feeding your fed by your glucose only. Now coming to the exclusion here, your fetal heart. Your fetal heart, it is going to be feeding glucose again. Your fetal heart will also feed your glucose. Now in an adult, when your heart is going to feed glucose, what can you diagnose? Remember, only your failure heart or your heart failure. So only in the heart failure at fed state, your heart will be taking glucose. Apart from that, always it is going to be fatty acid, which is going to get utilized in your heart. Now coming to the fasting state. Now I always told you in fasting state you have your fat involvement. So remember, so all your vital organs like your heart, liver, muscle, adipose tissue, they will be consuming your fatty acid. But remember, your fatty acid cannot cross your blood-brain barrier. Okay, cannot cross your blood-brain barrier and they can enter into your brain because I told you already, chylomicrons are the one that is going to take your fatty acid and going to supply each and every place. Now remember, proteins cannot bypass your blood-brain barrier. Therefore, you don't have any transporter for carrying your fatty acid. Therefore, it cannot cross your blood-brain barrier. Therefore, again, it is going to be a glucose that is required for maintaining the blood supply to your brain. Similarly, RBC. There also, it is going to be the glucose that is going to be required for maintaining the nutrition to them. And next, you're going to move towards starvation. Starvation is going to be amino acid and your protein. Remember, your liver can feed on whatever given. So, amino acid, but remember, your brain and heart, they cannot be because proteins cannot cross. Therefore, your brain level, it is going to be your ketone body that is going to be involved. And in your heart also, it is going to be a ketone body that is going to be helping you in maintenance of your starvation, nutrition. That is the reason why in the prolonged fasting state, there is going to be a production of acetylcholine. There is going to be production of acetylcholine by beta oxidation of your fatty acid. Similarly, your muscles will be feeding on your amino acid. Your adipose tissue, muscles will be feeding on amino acid okay, muscles will be feeding on your fatty acid, okay, and fatty acid as well as your ketone body, whereas adipose tissue will be feeding on your fatty acid. And remember, your RBC here also require glucose. So from this, what you can understand, your RBC is entirely dependent upon glucose. Your RBCs are entirely dependent on your glucose. Clear. The same thing I have explained here also. So brain, skeletal muscle, cardiac muscle, adipose tissue, I have given you in a shorter format. You can remember. Okay. Now moving on to discuss about the scenes in the fasting state. So what happens in the fasting state, guys? So during the fasting state, what happens? So during the fasting state, there is going to be a, this I already I have just drawn and I have just explained you. The same thing is present in this also. Now coming to the question, let me see who is going to answer. A 32-year-old on trekking expedition was stranded in the forest without any food supply, but was rescued after a week. So after a week now, it is going to be prolonged fasting, prolonged fasting. On medical examination, the plasma showed very high level of certain compound, which were the major source of energy for his vital organs. The main factors resulting in the formation of these compounds are decreased level of circulating glucagon. No, glucagon will be raised in the fasting state. Appa idu false. Decreased formation of acetyl-CoA in the liver. False. Acetyl-CoA is produced in the fasting state. So that is also false. Inhibition of beta oxidation. False, because acetyl-CoA is produced only by the beta oxidation. So increased level of free fatty acid in the blood, okay, is true, because all your triglyceride gets converted into your free fatty acid, and this free fatty acid will be converted into acetyl-CoA by the presence of acetyl-CoA by the beta oxidation pathway, from the beta oxidation pathway. So the answer is option D. Any doubt in this? Any doubt in this? Next, coming to the next one, acetyl-CoA is going to be a common intermediate formed from the metabolism of carbohydrate, protein, lipid metabolism under fed and fasting. Which of the following is a major source of acetyl-CoA under fed state? So remember, about your acetyl-CoA, add few short points, okay, because you're going to read about acetyl-CoA everywhere. In your carbohydrate metabolism, you will be learning. In your fatty acid metabolism, you will be learning. So you have to understand what is acetyl-CoA. Remember, acetyl-CoA is a 2C compound. So this acetyl-CoA give rise to your ketone body. This acetyl-CoA is required for cholesterol synthesis. This acetyl-CoA is required for your triglyceride synthesis. Okay, this acetyl-CoA is nothing but they are going to be a carrier of your TCA cycle. They are going to be intermediate of TCA cycle. They are going to be a substrate. They are going to be a substrate of gluconeogenesis. Gluconeogenesis. And very important, they will be activating. They are going to be activator of first step in gluconeogenesis. Gluconeogenesis. Remember, okay, in a fed state, you're going to have what? So the question given here is going to be fed state. Fed state now you will be having a carbohydrate available in your body. So this carbohydrate is going to get broken into two products. One, it can get broken into pyruvate, or it can go into acetyl-CoA. Acetyl-CoA. This acetyl-CoA is the one that helps in formation of lipid, or they are going to help in formation of protein. So you're going to have only pyruvate. So pyruvate converted into acetyl-CoA in the presence of pyruvate dehydrogenase complex. That is going to be your link reaction. So in the pyruvate, it is going to be the major source of acetyl-CoA. So pyruvate is a major source of acetyl-CoA. Everyone, is clear? Pyruvate is a major source of acetyl-CoA, because your pyruvate in the fed state, you're going to have carbohydrate, which is going to give you pyruvate. This pyruvate gets converted into your acetyl-CoA in the presence of pyruvate dehydrogenase complex. Okay, next, coming to the next question. A patient with a type 1 diabetes, okay, presented with the complaints of polyuria. Which of the following normally occurs in the body? Can you tell me which occurs normally in the body? Whether glycogenesis occurs in the body? No, because insulin is deficient here. Since insulin is not there, glycogenesis cannot occur because it is an anabolic reaction. Protein synthesis, again, it is an anabolic reaction, so it does not take place. So decreased cholesterol synthesis. Okay, so decreased cholesterol synthesis. Is it true? Will there be a decreased cholesterol synthesis? Will there be a decreased cholesterol synthesis? Anyone? How will be your cholesterol synthesis? It's a cholesterol synthesis is an anabolic reaction or catabolic reaction. Of course, it's an anabolic reaction. Therefore, it is not going to be occurring. So there is going to be a decrease in the cholesterol synthesis. So there will be a decrease in the cholesterol synthesis. Finally, what happens? Increased conversion of free fatty acid, fatty acid to acetyl-CoA. Fatty acid to acetyl-CoA. When do you have this conversion? Breakdown of your fatty acid. So here there is going to be breakdown of fatty acid. So it's a catabolic reaction. Now you have a confusion. Here they are giving decreased cholesterol synthesis. So they have decreased cholesterol synthesis. Here they are telling increased conversion. If you're going to see here, cholesterol synthesis நடக்காது. Decreased ன்னு சொல்ல முடியாது. Cholesterol synthesis நடக்காது. So if you are going to compare this option C and option D, your option D will be a perfect answer than option C, because here beta oxidation is going to occur. So increased conversion is going to occur. So if you're going to ask me which are the following, option C or option D, or which is right answer, I'll tell you that option D is the right answer. Clear? Option D is going to be your right answer. So option D is going to be your right answer. Clear, right? So any doubt here? So again, we will see another important concept. So we are going to see a concept called as respiratory quotient. What is this respiratory quotient? Respiratory quotient is nothing but CO2, which is going to be, so we are going to talk about your CO2, okay? So CO2, which is going to be produced, divided by oxygen, which is used. This is nothing but your respiratory quotient. So carbohydrate is nothing but, for carbohydrate, remember, the amount of CO2 formed is 6, and amount of oxygen utilized is also 6. So you will be having your RQ equal to 1 for carbohydrate. But for fat, what happens? It is going to be 0.7. So 0.7 now, okay, it's going to be 0.7. So that means that if you're going to produce 7 carbohydrate, 10 oxygen is used. 7 carbohydrate is produced, but carbon dioxide is produced, but 10 oxygen is used. For your protein, it is going to be around 0.8, around 0.8. Okay, so it's going to be around 0.8. That is going to be 8 carbon dioxide molecule produced, but only out of 10 oxygen which is used. Okay, so in your mixed diet, if you're going to go for mixed diet, it is going to be 0.85, 0.85. And if you're going to consume an alcohol, it is going to be 0.66. So quotient will be 0.66. Okay, quotient will be 0.66. So normal respiratory quotient will be 1. Okay, in diabetes, what happens? RQ quotient will be decreasing. That means even for carbohydrate, RQ quotient will be only 0.7. So for 0.7 for carbohydrate, even for a diabetic individual. Okay, so on giving insulin, what happens? Now we are going to give insulin. So while giving insulin, what happens? Now that is going to cause increase in the respiratory quotient. That causes increase in the respiratory quotient. Now, what are the things that increases your respiratory quotient, and what are the things that are going to decrease your respiratory quotient? So the one that is going to decrease your respiratory quotient is going to be a diabetes, fasting state, your starvation, and your alkalosis. Alkalosis. Similarly, when you're going to do a exercise, they are going to increase your respiratory quotient. Fever is going to increase the respiratory quotient, and acidosis is going to increase the respiratory quotient. So let me see who is going to answer this question. A 20-year-old female likes to eat a meal exclusively consisting of white bread, jam, and a big bowl of pasta. So bread, jam, pasta, இது எல்லாமே என்னது? They are going to be carbohydrate. On the morning of biochemistry of your biochemistry exam, she has overeaten the breakfast as she was feeling stress. So now what happens? Whenever you're going to take a carbohydrate, excessive amount of carbohydrate, that too, what happens? So normal carbohydrate, what happens? You have a respiratory quotient of 1. But when you're going to consume excess amount of carbohydrate, your respiratory quotient will be more than 1. So the answer here is option B. The answer here is option B. Clear? The answer is option B. Now we are going to move into a very, very important topic that is going to be your, shall I move into the next one? Yes. So we are going to see here that is going to be regarding your diet. So regarding the diet. So now you are now a doctor. So now you are a first year MBBS student. Now what happens is your friends will be asking you, Macha, I want to do a intermittent fasting. Your school friends will be asking you. So how to fast? So basically their question will be how to fast, how to reduce the weight. So அதுக்கு தான் மெயினா நம்ம fasting பண்றோம். Okay, remember 60 to 70% of our food is going to be consist of carbohydrate, and 15 to 25%. So remember the energy which we are getting, 60 to 70% is going to be from carbohydrate. 15 to 20% through food, we are going to take a fat. This fat will be converted into, these fat are going to be exogenous fat. So these fat are going to be exogenous fat, which is going to be consumed in your diet. So the food which you are going to consume, they are going to have 60 to 70% carbohydrate, 15 to 20% it is going to be of fat. And this fat is going to be exogenous fat consumed from outside, and they are going to be mainly taken care by your chylomicrons. They are going to be transported by chylomicrons. But the carbohydrate which you are going to eat, this carbohydrate, 50% they are going to be giving you energy. The remaining 50%, they will be like stored. The remaining 50% is going to get stored. Out of this remaining 50%, 10% is going to be glycogen, and remaining 40%, they are going to be stored as fat. This is going to be your endogenous fat. Remember, okay, so the exogenous fat is going to be taken care by your chylomicrons, whereas your endogenous fat are going to be stored as LDL, VLDL. They are going to be stored as LDL, VLDL, and they are going to be stored as your cholesterol. Now tell me which is dangerous, whether it is going to be your exogenous fat or your endogenous fat. Can you tell me which is going to be dangerous for you, exogenous fat or endogenous fat? In the chat box, please. Whether it is going to be your endogenous fat or exogenous fat, which is going to be dangerous. Remember, guys, exogenous fat will be utilized as free fatty acid by the. Can you tell me when you're going to eat a food, which is going to utilize your fatty acid, your heart? You're going to have your heart, which is going to utilize all your exogenous fatty acid, which is going to be traveled by your chylomicrons. But the endogenous fat is not utilized by your art also. So now what happens? It is just going to store as a kachada or a garbage. It is going to store as a garbage. Okay, so that is why whenever your patient is going to be, or your friends is going to ask a diet plan, okay, for weight loss. For weight loss, your AIMS protocol gives you as Atkins diet. ATKINS, Atkins diet. So this is recently asked in the INICET exam. INICET exam, they asked you this question. So after your final year exam, after your internship, you may go for your post-graduation entrance exam. So most of the questions that are appearing in your CBME university exam papers as a MCQ, they are actually taken from your previous year papers of your NEET, INICET, AIMS, PGH, Chandigarh, etc. That's why I'm taking those questions and discussing with you. So initially they ask you this Atkins diet. What is this Atkins diet? It is nothing but low calorie, low carbohydrate diet, low carbs diet. Okay, wow. So now the question to you: can you consume honey and fruits? Fruits, juices, especially fruit juices, while you're going to have a patient who wanted to reduce the weight. Can you suggest eating honey? Can you suggest eating fruit juices while when the patient wants to reduce their weight? Of course, remember these honey, fruit juices, everything are going to be fructose. Okay, this is the thing I want to tell you. Your WhatsApp university tells you that the WhatsApp information that is shared by the elderly people in the one that are going to be shared by the elderly people in your WhatsApp groups, okay, your family groups, they tell that you can consume honey, you can consume fruit juices for reducing the weight loss. But if you want your friend to truly reduce the weight, I'm not talking about any other things. If you want your patients to reduce weight, you should now ask the patient to avoid fruit juices. You want to ask the patient to avoid honey because scientifically they are going to be fructose. Remember, your fructose, your glucose, everything can just convert to produce lipids, or they are going to be lipogenic in nature. So ultimately, you want to lipolyze your patient, and if you're going to ask your patient to, you can just give a permission to consume honey, fruit juices. They are going to be again lipogenic in nature. So this lipogenic nature will be increasing the fat of the patient. It will never decrease the fat of the patient. So with regards to the weight loss, you have to always be sure that tell your patient that fruit juices, honey, they are not going to reduce. Morning, காலையில எழுந்து the patient will be telling you, sir, morning நான் ஒரு தண்ணியில honey ஊத்தி குடிக்கிறேன் சார் வெயிட்டை குறைக்கணும்ட்டு. Remember, honey சேர்த்தீங்கன்னா they are fructose. They are not going to reduce your weight. They are not going to reduce your weight. Remember, they are lipogenic in nature. Okay, lipogenic in nature. So that you need to remember. And second thing I want to discuss you is thermogenic effect. Thermogenic effect, or otherwise called as special dynamic action. What is this thermogenic effect or special dynamic action? Remember, you require some amount of energy. Okay, amount of energy required to digest or absorb, transport, and metabolize your food. So and the energy, okay, is going to consume some amount of molecules which you are going to take, which the patient will be taking. So this is called as thermogenic effect, otherwise called as your special dynamic action. So otherwise you're going to call it as a special dynamic action. So this is a very important point you need to understand. Okay, this is a very important point. You're going to understand what is special dynamic action. Special dynamic action means thermogenic effect. So you require some amount of energy given by some molecule. You have some amount of energy given by some molecule which is utilized just for the production and transport. Okay, just utilized for the production and transport. So which are the molecules that are going to be utilized by your body for the production and transport? So that is going to be, so your carbohydrate, many people will think, but it is going to be a protein. So the amount of protein you're consuming, they are going to give some ATP. From that only you're going to have the energy which is going to help in digestion, absorption, transport, and metabolize, and least comes from your fat. This is called as specific dynamic action. This is called as your specific dynamic action. So this is called as your specific dynamic action. Next, what you are going to remember is a very important thing, that is going to be your DASH diet. DASH. What is this DASH diet? Dietary approach to stop hypertension. So this is another diet which they can ask apart from your Atkin diet. What is your DASH diet? Remember, DASH diet is they are very much having a less amount of sodium and increased amount of potassium, calcium, and magnesium. So this I want you all to remember. This is something about the basics of your diet. So maximum people வந்து பாத்தீங்கன்னா இந்த பயோகெமிஸ்ட்ரியோட நீங்க டயட்டை பத்தி மறந்துடுவீங்க. That is why a major complaint is there on doctors that they don't know about the diet prescription. So why? Because we are not actually reading about this diet except from your biochemistry. Remaining all the places we don't read why you wanted to give some specific diet for the patient during specific times. Okay, that's why I'm explaining about this in the basic concept. Next is going to be something which you are reading from your 12th, 11th days, NEET UG days, you are reading about your rRNA. You have your, this is going to be your endoplasmic reticulum. This is going to be your endoplasmic reticulum. This is going to be your nucleus. Then you're going to have your nuclear pores. This is going to be your chromatin, nucleolus, nuclear envelope, etc. Then you're going to have your mitochondria. So mitochondria, inner membrane is going to be semi-permeable. They are going to be semi-permeable. That means example is going to be a cardiolipin. Cardiolipin is a molecule that is going to be going through this. That is a semi-permeable in nature. Next is going to be your peroxisome. So this is going to be a peroxisome. This peroxisome, they are going to be, what are the pathways which is going to be taking place in your peroxisome? Number one, you're going to have your alpha oxidation. இது வரைக்கும் நீங்க பார்த்தது எல்லாமே beta oxidation. You have something called as alpha oxidation, which we will be discussing in your lipid biochemistry. Alpha oxidation is going to occur in your peroxisome. Second thing, you are going to have your breakdown. So if you are going to have your breakdown of toxic products. So breakdown of toxic products is done by your H2O2 molecule. That is again done by your peroxisome. Remember, what is the difference between a beta oxidation of fatty acid and alpha oxidation of fatty acid? In alpha oxidation, you don't have production of ATP. Whereas your beta oxidation, there will be a production of ATP. That difference. Next, coming towards your next one, that is going to be a peroxisome. Remember, peroxisome, you're going to have this H2O2, which is going to break down your break catalase enzyme to form H2O and O2. So they are going to neutralize your toxic element, free radical. Neutralization of free radical is going to be taking place in your peroxisome. Now coming to the next one, lysosome enzymes. So lysosomes are nothing but they are acid hydrolase. So you're going to have acid hydrolases. So what is this acid hydrolase? This acid hydrolase, they are going to help in breaking down. They are going to be helping in breaking down your macromolecules. Again, they are going to be involved in breaking down of macromolecules, breaking down of your free radicals. So they are going to help in breaking down of your free radicals, okay, toxicity ல இருந்து prevent பண்றதுக்கு இவங்கெல்லாம் help ஆறாங்க. Now coming to the liposomal storage disorders. You have something called as lysosomal storage disorders. What are these lysosomal storage disorders? So what happens basically is you have some storage. This lysosome is going to actually store some product. Now what happens whenever you're going to have, okay, whenever you are going to have this enzymes, this lysosomes not functioning well, what happens? Your lysosome will be starting to accumulate the waste product, but this waste product will not be broken down by your acid hydrolase. You require acid hydrolase. If acid hydrolase is not available, what happens? There will be just storage of waste product inside your lysosome instead of breaking down of waste product. So instead of breaking down of waste product, this is called as lysosomal storage disease. So you have something called as lysosomal storage disease, where the lysosomes are unable to break down the waste products. There are going to be accumulation of glycosaminoglycans like your mucopolysaccharides. They will be getting accumulated. That is called as your lysosomal storage disease. Okay, so we will be discussing about the size cell disease. We will be discussing about the Pompe's disease. We will discuss about the cystinosis, sphingolipidosis, and Wolman's disease in the future. Don't worry, we will be discussing. For now, just remember that these are going to be problematic. Then coming to your okay, endoplasmic reticulum. So remember your endoplasmic reticulum. So I'll tell you what is this eye cell disease. Remember, eye cell disease is nothing but inclusion body accumulation. So for now, just write it down like this: inclusion body accumulation. Pompe's disease is otherwise called as glycogen storage disease type 2. So where you're going to have a acid maltase enzyme deficiency. Acid maltase enzyme deficiency. Okay, then you're going to have sphingolipidosis. So example is going to be a Gaucher. So example is going to be a Gaucher. Remember, this is the most common lysosomal storage disease you have. Then you have Waldman's disease, where you're going to have your acid lipase enzyme deficiency. So acid lipase enzyme deficiency. Clear, right? Now we are going to move towards the next organelle, that is going to be endoplasmic reticulum, mainly involved with protein synthesis, Okay, you're going to have rough endoplasmic reticulum and smooth endoplasmic reticulum. Why do you call this as rough endoplasmic reticulum? Because they are going to be along with ribosomes. Okay, now coming to the next one, Golgi apparatus. Golgi apparatus is otherwise called as gift packer. So you're going to call this as gift packer because they are going to help in protein packing. So storage, phosphorylation, tagging, segregation, and etc. So there's going to be segregation, etc. Now you have some disease called as protein targeting disorders. இதில் என்ன ஆகும்னா இந்த protein synthesis normal இருக்கும். Protein synthesis will be normal, but protein packing by your Golgi apparatus will be abnormal. Okay, so because why? Because what happens? Because of the abnormal packing, they will not go to the target areas. They will not go to the target areas. That is called as your protein targeting disorders. For examples, in case they are going to ask in your Viva, you should be able to know: eye cell disease, primary hyperoxaluria, primary hypercholesterolemia, Zellweger syndrome, and cystic fibrosis. Don't worry, we will be discussing about all these things in detail. Okay, we will be discussing all these things in detail. Zellweger syndrome mattum, here the problem is present in the peroxisome. Peroxisome liye problem ஆயிடுது. Because peroxisome will be packing a empty cell. So an empty cell it is going to produce. So that is a problem with your Zellweger syndrome. Don't worry, we will be discussing. Next comes your ribosome. Ribosome is going to be mainly forming your organelles. It is going to produce your DNAs. So that is going to be a problem in your ribosomal disorders. Now coming to the last topic of the general biochemistry, first chapter of your Vasudevan, that is going to be your cell organelles and your marker. Remember, for plasma, it is going to be sodium, potassium, ATPase, plasma membrane. Then for Golgi apparatus, it's going to be galactosyl transferase. Cytosolic, it's going to be lactate dehydrogenase. Ribosome ka rRNA, endoplasmic reticulum ka glucose 6 phosphatase, peroxisome ka catalase, nucleus ka DNA RNA polymerase, mitochondria it's going to be succinate dehydrogenase, glutamate dehydrogenase, and for lysosome it is going to be acid phosphatase. So with this, I'm completing the first part. Now, okay, first part of discussion of entire discussion of us, that is going to be your general biochemistry. So in general biochemistry, the last and final thing which I want to discuss with you is about your cell membrane. The last and final thing I want to discuss with you is going to be a cell membrane, just 10 more minutes. Okay, can you tell me Okay, can you tell me one thing? Okay, what is the law of thermodynamics? விழுந்து படிச்சிருப்பீங்க. Do you all remember the one thing which you have been reading from your, or I'll give you this homework. Okay, so let me do one thing. So all these you're going to revise and come back tomorrow. Again, tomorrow we are going to have biochemistry session. At that time, I'll be asking you these questions. So what is thermodynamic? What is laws of thermodynamic? Law of thermodynamics. Take your physics book or anywhere, wherever your book you have. So from your 12th, you're going to learn and you're going to tell me what is law of thermodynamics in the next session. Thermodynamics in next session. Okay, so you are going to tell me what is the law of thermodynamics. So what is going to be this G? So this is equal to H minus delta S. You would have been reading this, right? And you're going to tell me what is exothermic reaction and what is endothermic reaction. Endothermic reaction. So you're going to tell me all these things, and you will also have in your UG NEET, you would have read this Donnan membrane equilibrium. Donnan membrane equilibrium. Membrane equilibrium. So in the 4 point, you're going to read and come in the next class, so that I will tell about why I was asking you, and I will tell you, and I will move towards the first topic, that is going to be the enzymes. So today I'm not taking ETC. This itself is enough. 1 hour session is more than enough for you all. Very good. Laws of thermodynamics. The first thing I think Xiaomi is telling, the energy can neither be created nor be destroyed, but it can get converted from one form to other form. Can you tell me the second law of thermodynamics? There are 3 laws of thermodynamics. First law, Xiaomi has told. The second law and third law, you're going to find and you're going to write it down and come in the next session. Tomorrow at 6:30, we will have the tomorrow session. So at that time, I'll be discussing with you about these 4 things, and let me move towards the enzymes. Let me discuss about the enzymes. Clear? Thank you so much for attending. Just put in the, you can just, I'll be sending you a very important. I'll be sending you a Google form. I request you all to kindly fill that Google form and tell me every session, after every session, you're going to fill that Google form and let me know how the classes. If you need any improvement, I'll just do this. If you want any suggestion, you can just let me know every day. Either it is going to be a live session in Zoom, or I'll be doing a session in the YouTube. But anyway, you will have session. Okay. So don't worry. Thank you so much.